feels like science fiction turned real.
So we take cells from a patient, genetically engineer them, and put them back into the patient, recognize the tumor again, and kill off the tumor cells.
That sounds magical to me. Henry Erdlei today, who is a doctor, scientist, and an entrepreneur, driving GoCART. CAR-T stands for chimeric antigen receptor T cells for cell therapy, specifically for cancer patients. So what are the cell therapies that existed till now in the market, and how is GoCART changing that?
The problem with CAR T cells is you have to choose a target antigen, and what we do is a so-called AND gate. While killing everything that has CD56 is pretty deadly, we might use it in a combination.
Hi everyone, welcome back to Build with Shreya, where ideas and stories inspire you to build something extraordinary. I am in conversation with Henry Erdlei today, who is a doctor, scientist, and an entrepreneur. And here they're looking into innovative methods of cell therapy for AML, which is acute myeloid leukemia, as well as multiple myeloma. So today is not just a tech talk. We are here to talk about life impacting innovation that is happening in the world of cancer treatments, where cell therapy is set to become safe and more accessible to patients across the world. So let's dive in and see what's there to discover. So, Henry, what impresses me about cell therapy is how it is changing from like a one size fits all kind of approach to a more personalized and more magical treatments. But it is so new, though, it feels like science fiction turned real. So, can you walk us through how CAR based cell therapy is like a game changer, especially for cancer treatments?
CAR-based cell therapy, most people, if they know it as CAR T-cell therapy, is a type of, we call it adoptive cell therapy, adoptive immunotherapy, which means that in broad general, immune cells are transferred to a patient. So how does the CAR system work? CAR means chimeras antigen receptor. So what we do is it's mainly used for cancer patients. And problem with cancer is if someone has cancer, then the natural immune response, the normally NK cell and T cell response that kills off aberrant or somehow sick cells, like cancer cells, must have failed. So the cancer had the chance to grow out and grow into a tumor. And what we do is we take immune cells from the patient, and then we genetically engineer them with this CAR, with this chimeras antigen receptor, an artificial receptor that these cells can use to recognize tumor cells. And then this activates the cells and kills them off. So we take cells from a patient, genetically engineer them, and put them back into the patient. And with this artificial receptor, they then home around the body like normal immune cells and can now recognize the tumor again and kill off the tumor cells.
So, I mean, that sounds magical to me. But could you also tell us about the challenges that are there while executing these kind of therapies, and how have you planned to mitigate them through GoCART?
Yes. So there is on one side the challenge of the tumor microenvironment, especially within solid tumors. So if we look at these cell-based immunotherapies, by now we got approved regimens for hematologic tumors, diverse sets of B-cell lymphomas, the ALL, and the multiple myeloma have approved CAR T-cell therapy. It is kind of difficult to get into solid tumors. Why is that? Solid tumors grow a specific set of cells around them. They create kind of a tumor-specialized organ, the tumor microenvironment, where they change their surroundings to benefit the tumor cells, which often includes immunosuppressive cells and agents being around there. and the whole environment being hypoxic, which is also suppressive to immune cells. So therefore, it's kind of difficult to get into there with the cell therapy and be really effective, unless you use tweaks to make the immune cells resistant to that or even activate by that. So there is approaches like hypoxia switches that can make them more alive, make them more aggressive inside this hypoxic environment, where you turn the hypoxia signal actually into a proliferation and not a stop signal. And also there is regulatory T cells the tumor attracts that tune down immune response via specific pathways. But you can also interfere with that and try to block these tricks in recent trials with CAR T cells. So we're getting there within the last steps.
So, what are the cell therapies that existed till now in the market, and how is GoCART changing that?
Yes. So by now we got different regimens for two kinds of antigen. We got CD19 and BCMA CARs. So CD19 CARs are against the antigen CD19 and are used originally in ALL, but currently they're mostly used in diffuse large B-cell lymphoma and can be used in diverse B-cell lymphoma classes. And then we have the BCMA CAR, which is used in the multiple myeloma. So what actually does GoCART do? If you look at these antigens, they're really specific, and they are chosen for a reason because they are so-called druggable antigens. So the problem with CAR T cells is you have to choose a target antigen, so-called an antigen is a surface marker, a protein on the surface of the tumor cell that identifies the tumor cell, but is not on a healthy tissue you desperately need to live. So, for example, the BCMA CAR is against this BCMA antigen on the multiple myeloma, but this is actually maybe the second or third best antigen on this type of tumor. Way better would be an antigen called CD56. And this is hugely expressed, and it can't be downregulated like BCMA because it's one of the big growth driver of this type of tumor. So the question is, why don't we have this as a CAR T cell instead, or at least in addition to BCMA? And this, it is not druggable. Why is it not druggable? It is on the multiple myeloma and also on other types of tumors like the AML and glioblastoma, small cell lung carcinoma. But it's also on every neuron in your brain.
Okay.
And killing off all those is kind of counterproductive.
Absolutely, yeah.
So GoCART now is about the question, how can we use this in a safe way? And what we do is a so-called AND gate. While killing everything that has CD56 is pretty deadly, we might use it in a combination. So, for example, the AML, acute myeloid leukemia, has high levels of CD56 and also expresses CD33, a blood cell specific antigen. And in nature, they're not together. They're not together on the same cell, but they are together on this aberrant on the tumor cell. So what we want is a CAR T cell that kills off a cell only if it has both those antigens and not only one, so-called AND gate. So a bit similar to an AND gate in coding. You need both signal to get the reaction. And if you can manage that, then you can selectively kill off the tumor and nothing else. And this is also important safety layer for existing antigens, because like they come at a trade-off. So you kill off specific types of B cells when killing off everything that has CD19, or the right plasma cells if you kill everything with BCMA. So there are some downsides. It's just not so tragic that you wouldn't do it to get rid of cancer. But even those would profit from this kind of mechanism. So we looked into this, and past approaches sadly failed pretty much. They delivered kind of if better gates. So they are a bit leaky. They tune down killing a bit, but not enough. So people have tried having one receptor for antigen A that then expresses the second receptor, and that is the one that kills, or split up the receptor with different stimulatory domains that are intracellular, and have them in separate receptors so that the high signal is only possible with both of them. But even if only one of them is there, you get a weak signal. And the weak signal is lower on killing, but it will still kill off the target cells. So if you use CD56 with these kind of systems, you will still kill off your brain, only slowly. Mm-hmm. So not a solution for this kind of problem.
So how do you go about having these combinations for antigens that can kill the right target cells and not anything else?
So you can look at libraries of antigens and of tumor antigens. The Cancer Genome Project is a good source for that. And sequencing data is getting more and more accessible. So you can find the combinations there. Mm-hmm. And then we at GoCART actually moved the whole system extracellularly. So in order to have a full signaling, we have a common trunk on the CAR T cell that is now the same CAR, the same receptor for whatever antigen and whatever cancer you want to target with our GoCART system. And these combination antigens get specific AND gate double binders, how we call them. Mm-hmm. So this is another small peptide drug that is administered. We aim it subcutaneously, and that just swims around in the body and binds these antigens. Mm-hmm. And they come in two different types, A and B type if you want. Mm-hmm. And if these two different types are together in the same cell, they can form a dimer, they bind together. And this dimer is actually what our CAR binds to. So our CAR then recognizes the cells that have both binders on them, mm-hmm. and kills them off. But a single binder has no affinity for the CAR. So the CAR does not kill off this specific cell. So we get a real AND gate that does not kill off your brain. And we still get the full signaling cascade. So another problem with an attempt was to intracellularly to do an AND gate where they succeeded at the AND gate, but then the cytotoxicity was low. So they really needed a lot of signal, and a lot of signal means you need a lot of antigen, a lot of these proteins on the surface of the tumor cell to make the cell kill it. Mm-hmm. And for that purpose, this will be a problem because cancer cells are known to downregulate, mm-hmm. in response to therapy. They will tune down their expression of specific antigens to a minimum, and then this approach would fail to recognize and kill them off. So the cancer can easily escape that. And to be a really effective therapy, you need to work around this. And therefore, we wanted a whole CAR inside the cell with all the possible stimulatory domains in there and the full potential signal to have the lowest amount of antigen necessary to kill. But we still wanted the AND gate. So we moved it extracellularly with this type of binders.
Great. So, I'm sure you worked on researching all of this extensively before. So tell us a little about that journey. What kind of breakthroughs did you come through while you were doing the research? And then how did you identify this as an opportunity? What gaps did you see apart from the technology, just in the market of the solutions that were existing for cancer treatments? And what was your motivation?
Yes, that actually was a me and CAR T-cell therapy date way back in the interest. So actually got interested in CAR T-cell therapy through a presentation I did at high school. So it has been back of my mind for quite a long time. And then I started studying medicine. And then I remembered, well, I really like this tech. This was really cool, and I wanted to do something serious with it. So I looked for doctoral thesis in the field, and then got introduced to working with the CAR NK cells. What is CAR NK cells? NK cells are another type of cytotoxic cells. So we got T cells and NK cells. T cells are specific. Normally, they have a specific receptor for one kind of antigen to kill that one off. And NK cells are more diverse. They're just homing around your body and looking for cells that just are a bit weird, send stress signals, downregulate MHC, which is a molecule they present stuff on. So they just look for cells that are off and kill them off them. And you can do CARs with both. So NK cells have the advantage of being transferable cross-person. So if you want to transfer T cells, it would have to be matched like a stem cell transplantation, which is pretty hard to find. So T cells is mostly done autologous, so they're taken from the patients themselves. But NK cells, in theory, could be used as a donor product or even as an off-the-shelf product with a cell line. Have a shorter half-life, so there is a bit downside, but their big strength may be at points where expanding T cells fails in the patient. Because you have to know that patients who get this kind of treatment, they normally have received several lines of chemotherapy, and a big chunk of them has bad quality T cells that fail to expand, and you can't make CAR T cells out of those. So NK cells are good options for that one. So I looked into that one and did a BCMA CAR again, and had a homing improving project where we expressed so-called cytokine receptor, that chemokine receptor that attracts cells into the bone marrow niche to make them hunt the tumor better, if you want. Then looking at the data of around this and the search for antigen, the problem was pretty clear with this antigen scarcity. So it is always about finding an antigen that could be used for this type of therapy, because of the problem of we call it on target of tumor killing. So CD56 would kill your brain. BCMA, okay, it can be used, kills off plasma cells. Worst case would be the patient will have to substitute immunoglobulins, so antibodies, for the rest of his life, but you can live with this. It is go to hospital once a month and get an infusion. It is better than dying from cancer, but still not ideal. So this antigen hunt was a real problem, and the idea of AntGate has been around for quite some time, and there was these intracellular approaches that kind of failed. So I had the idea, why not do it extracellularly and do it like in GoCART. Difficulty was how to make this kind of sequential trimerization, because it's kind of a complex step to have these binders that we inject, float around, and then we want them to bind together first before being able to bind to the CAR, which was kind of hard to engineer. But like 3 years ago, a type of peptide was published that could be used to do this. So this is how we I decided to look for my co-founders and start off this kind of project. So I actually met them through iGEM Startups 2024. So iGEM International Genetically Engineered Machine Competition is what that means. And they have a startup incubator branch where just started proposing the project and found very engaged, very clever people to do this with, and this is how this became a company and very serious project in the end.
That's amazing. I mean, sort of, it's fascinating how a high school presentation can turn into something that becomes a motive and goal to sort of go out for and then build a company out of. But tell me about the different challenges you're facing as you're having this startup go live, and what are your next steps in terms of research, in terms of deployment? Do you plan to do any pilots? Like, what is the next step of action that you plan to do?
Yes, so the next big step is doing the in vitro work, of course. So by now, also because we needed time to look for funding, we were only able to do in silico work, which means we simulated and we keep simulating different candidates of these trimerizing part, the core technology of GoCART's type of CAR T cell, to assess their binding probability, their binding ability, and predict the best candidates to put through tests in vitro. And now we got a collaboration with a university in Spain, the University of Cadiz, where we will go and then run those candidates through an in vitro pipeline where we actually express these as a protein and assess their binding in a process called SPR, a laboratory-based binding assay. And good candidates from there go to a specifically tricky process that is a specialized FRET, fluorescence-related electron transfer experiment. This is where we actually want to express them in a human cell line, and then it's kind of an experiment where you've got these three parts and link them to fluorescent proteins that can shine in specific colors. And we use a trick that if they are bind, they can engage each other, even though they normally wouldn't be engaged. So we can shine at one of those, and if they're bound, this one makes the others shine as well, even though we don't put light into the experiment that makes the others shine normally. So if the other ones shine, we see that they are definitely bound, and that this is not degraded by human proteases. This is why it needs to be in this type of cell in the first place, because this type of peptide is being published to be sometimes broken down by human proteases, so proteins that just break down proteins and recycle them. And that would, of course, make them pretty useless as a therapeutics if you just break them down, even though they might bite. So therefore, we need this experiment as a mediator to predict that one.
So how long before, cancer patients can actually take advantage of GoCART? What do you think is the roadmap? How many years do you think that will take? What are the kind of challenges you think regulatory framework will bring in? So what all is there in pipeline?
Yes. So, of course, regulatory framework will add some years to this. So, to find the ideal trimer and actually then design the first CAR T cell with it, this might, if it works well, only take a year. But of course, then we need to approve it. So we need to do a larger animal trial and then a phase 1, phase 2 clinical trials. And we already plan on taking a shortcut there because we want to exploit so-called orphan designation and orphan approval. This is a special jurisdiction many healthcare systems have, where you can approve a therapy after a clinical phase 2 without the phase 3, under the precondition that you solve an unmet medical need or it is for a rare disease. And we exploit this by offering treatment options for the AML, which has no cell-based immunotherapy or immunotherapy at the moment. And with this end gate, this gets possible by exploiting CD56. Yeah. So we make the undruggable targets druggable, and we could be the first ones. Therefore, we could get this type of fast approval. And also by offering something to CAR T-cell relapsed patients in the multiple myeloma, where we combine CD56 again, this time with CD38. Because, well, the BCMA CARs, compared to CD19, they underperform. So actually, after a few years, patients do relapse who got this. And we would add a new option to change the antigen even for them. So there still is the unmet medical need. And by leveraging these two types of tumor, we might get this faster approval. So we calculate now with an optimistic calculation. So 2032, 2033 may be possible to be on the market officially. The end of the 20s might be the beginning of a phase 1, phase 2 clinical trial, where patients could get the treatment inside the clinical trial. So we want to fasten the process, but of course there is several regulatory steps, especially a larger animal study to be done before we can do this.
So tell us about how you think can digitalization or the new age AI or Gen AI based technology can help in identifying the combination of binders or antigens going forward. Where do you think technology can help fasten the process?
So actually technology is already pivotal in our process. The trimeric part we are building in silico actually is kind of an iterative AI process. So we start from a rather unspecific primer and have specific constraints. And then we train a model that predicts binding. And we have targets of these binding interactions, AB first, ABC then, and no AC or BC, because that would be a single binder binding to the CAR, which of course is what we want to avoid. The model learns this, so it iterates through, spits out mutated strands, and then ranks them. So we get a new list of antigens, trimeric parts, and the best performers of this mutated version, then they are mutated again. And it selects again the best performers. And this is basically how the in silico process for us works. So AI already is pivotal to create these artificial binding domains, which would be very hard in the pre-AI era, actually. So without, we don't use AlphaFold, we use a commercially available variant of that, of course. But it would be very hard to design this kind of peptide in this way without engines like that. And it would take probably many years to do this by hand.
I mean, when I come across, use cases for AI and digitalization, they are less life impacting. And to see an example of how it is transforming the way treatments can be done for issues like cancer and therapies, I think it's amazing, to see how long-ranging impact it can have in bringing those therapies out, the right therapies out in a quicker fashion.
When people talk about AI these days, of course, most mean like the speech engines.
Yeah,
So
LLMs.
because LLMs, this is because it's the new thing. But well, things like AlphaFold, the protein prediction engines, that they actually have been around for a few years longer than the LLMs, and then they got kind of like forgotten, maybe because not much of them is really public facing. They're more for people, like doing this stuff like we do. The public can't do much with them, but they're actually really pivotal for drug research, actually. So they serve a big purpose in there. Developed by DeepMind, which is now a Google subsidiary, was like the first big protein prediction engine, and which is really useful and in predicting the binding behavior of proteins as well as internally, so sterical, how they look, but also externally in how they interact with other proteins. It's actually a very good tool for research and open source, but it's not allowed to use it commercially. So for using it commercially, like us, we need commercially available software that does the same. But AlphaFold was like really the first one that could deliver this, and is really good at doing it, and is in wide use in the research environment.
And with the rapid pace with how AI technologies are progressing, so platforms like these have also adopted them. Like, are they also advancing at the same pace, or are they still sticking to the methods they used earlier to predict different combination of binders and identifying proteins?
Well, they actually have been around for a bit longer than the LLMs, so they are not changing anything, changing everything every 3 months anymore. They're kind of like grown out of this phase and become really good by now. But you still see a lot of development around the libraries that are the foundation of those models. So we see more and more specific libraries adapted to specific types of proteins, specific use cases. Because like this is kind of like the underlying thing with with AI. So people say bullshit in, bullshit out. You need really good source data to get good predictions. And if you can adapt source data to your specific purpose, your predictions also get better for that specific purpose. So we still see a lot of development in what kind of data they use. Not really in the architecture anymore. So most of them use graph neuronal networks. But the underlying data is getting more and more specific and more vast, and tries to incorporate more and more peptides and sequence more and more peptides from nature and get their structure data.
Perfect. So let's get into more on the ground stuff, like distribution.
Yes.
So when we talk about GoCART cell therapies getting distributed, how do you foresee that? Would it be in collaboration with hospitals, clinics? What level of collaborations would you require to distribute the cell therapies to as many patients as possible in the most effective manner in the future?
This is also like one of the challenges CAR T-cell or adoptive cell therapy in general has, is a distribution. Because of course you have to handle this cell product. And this is kind of tricky business, and GMP production requirements make this even trickier. So maybe briefly how it is done by now. So by now, a cancer center, hospital, or ambulance is taking the T cells from the patient. The process is called apheresis. Is similar to a plasma donation, just this one collects the T cells, so the white blood cells, and the T cells are then singled out in a laboratory step that comes after that. And then these white blood cells are frozen down, submerged in a big bucket of liquid nitrogen, and then shipped around the world. So they're actually air transporting these buckets to the facility of the manufacturers, which are, by now they're all in the US. And then they get thawed, they get expanded, cultured, genetically engineered, cultured a bit more, frozen down again, sent all the way back to the hospital. Now 3 to 4 weeks have passed. And then they're given back into the patient for treatment, which is, of course, a process that is pretty lengthy and is expensive, of course, and actually not that good for the product quality, in fact, because freezing cells
again and then
always make some of them anergic, so they still live, but they won't do what you want. They just consume medium and that's it. They don't grow and they don't do their job. And others die. So what you want is you actually don't want freezing steps. But there is a number of developments that might in the near future change that. So one of them is in-house production units. Also what we aim for in the first step is that there actually are these closed loop machines that different companies offer. There is the Clinimax Prodigy from Miltony, and there's also the Hive from a competitor. They are trying to offer a system for the hospital to produce this in-house. So these apherosate, these collected white blood cells can just be hung on the machine, and the machine does all the other steps automatically. It takes the vector virus, so genetic engineering in this case is done by a modified virus that contains not how to build a virus, but how to build a car written inside. And the machine automatically takes the virus from the virus back and the cells from the cells back, and then it centrifuges them and transduces them. So the machine automatically produces the cell product that can then be tested for quality. You can produce the cell product in-house with this method, and then you save these 2 freezing steps. You do not need to freeze down the cell product, which means you don't lose a lot of T cells, which might still be bad quality because, a lot of treatment that preceded the CAR T cell therapy, chemotherapy, often radiation. So you don't make a probably already bad cell product even worse. And you save time, because in 3 weeks, especially for the ALL patients with the CD19 CARs, this really is a challenge. They might not have that kind of time. Normally, pretty much all patients for CAR T cell therapy need bridging chemotherapy to keep the tumor at bay, at least for the time in between cell harvest and giving the cells back. This period can be shortened as well, because you don't need the time for shipment. So you get a better cell product, and you get a shorter reaction time, which is actually the best outcome for a patient. If it fails, then we could switch to NK cells, or if it's prone to fail because of specific pretreatment, we get more and more data on that to predict that clinically. Then you could switch to NK cells, or already order NK cells at the first place. You can use donor NK cells from pretty much every human on another human. So they could be turned into some kind of semi-off-the-shelf product that can be used in there. And yeah, you also get down on complexity steps, which of course can bring down costs of manufacturing.
So tell us about this in-house production units, and probably you're suggesting that the hospitals can have them
Yes.
in their facilities. So in terms of costing, right?
Mm.
What does that mean for hospitals, and like how does the treatment cost for the patients eventually get impacted overall?
Yes. So the in-house production units is they're not yet GMP approved. These companies that build them, they are working hard to get them through regulators. In medicine, sadly, much of blocking is regulatory. But at the moment, one of these machines costs about $1 million on the free market. And so, of course, it's a big investment, and it's probably for the larger cancer center, larger hospitals. But on the other hand, with the methods as it is, CAR T cell therapy costs 400 to 500k euro per patient in European countries, and can go up to 800, 900k dollars in the US. And by getting down on these steps, and maybe also using the modularization from GoCART, as we can use the same viral vector for whatever antigen you can use. So economics of scale, standardization, these might offer to slash the costs in half, right, on market impact. So we could enter the market with costs of 200k per patient and drive it down while we scale up. So I think in the long term, by exploiting these in-house production units and maybe even in vivo CAR T cells, this is kind of the late big thing where you inject your viral vector directly into the patient and transduce like the T cells directly inside the patient. It still has some regulatory and safety questions, of course, but it might be one part in the future. I'm positive in the long term, CAR T cell costs can go down to the low double-digit thousands, which is manageable for most healthcare systems, I think.
I mean, you really gave a very clear picture into how the costs are right now and how they can be impacted with the new methods coming in. If you talk about entrepreneurship in general, it is challenging, right?
Of course.
I mean, there are so many moments of doubt, of question. And even if we don't talk about the complexity of the idea or the execution involved itself, you know, being an entrepreneur, even for a single idea or a simple business problem, is complicated in itself just because of the cycle it is, to sort of go through the entire process. But talking about your venture, GoCART, the the ideation, the research, the regulatory roadblocks, the execution, the implementation, the distribution, each step is a challenge in itself. Apart from all of it being an entrepreneur, so how do you deal with it? Right when you started thinking about these ventures, what were the low points that you went through? How did you keep up with the motivation, and what keeps you going?
Of course, in medicine, it's kind of like the maybe a bit more demanding game you have to play, and you have to want to do this. On the other hand, but the other hand, I think like motivation is pretty much there. You see, like when I think about every day, new medical innovation is stalled. Patients might suffer, and patients do die in that case. So what better purpose can there be in a business? So actually, how helping a lot of people and making a bit of money in the process. What better motivations can there be? And actually, well, by being a medical doctor, and that means I have seen the other side. So I have seen how the patients look and what kind of people are there, and what is the customer, and that this is something that is really needed and that could make many people's lives way better.
So, if you were to give a piece of advice to entrepreneurs who are newly entering the field of cell therapies, what would be your advice and what is it that they should be expecting in the first few years?
My advice would be persistence and real data backing, because cell and gene therapies are highly regulated, even more regulated from the production perspective than traditional small molecule therapies, as they are kind of new, and many regulators, they consider them kind of obscure. So to ease them, you really need to get production and your data right from the start, to avoid pitfalls of you need this experiment, we want more of this, and this can stall your venture by quite a lot. So what you should do is get neat data, get good assays that prove efficacy, already plan with assays for plasma half-life and some kind of safety assay before applying for clinical study, to avoid getting set back. Probably will still get set back by regulators, but you can reduce that to a minimum by looking at what kind of experiments are kind of common for your type of therapy. For example, killing experiments in vitro where you culture target cells, so tumor cell lines, together with the CAR T cells, is a common assay for CAR T cells. And even though you might have in your mind a better assay, it is sometimes better to stick with a common assay because regulators will want this data. And you should stick to that kind of data, or at least be able to explain very well why you made specific changes, because they will ask and they will want something comparable to what has been done in the past. And try to engage early, try to engage with hospitals early. Consider where you get your patient-derived samples, especially for cell therapies. For CAR T cell therapies, I need a steady stream of fresh T cells from donors. Attach yourself to a hospital or a university that has a license to obtain cell material for clinical study purpose or preclinical study purpose, to get your pipelines running. Think about animal trials early, because especially in Europe, applications can take a long time, and try to get someone in your environment who ideally already has an application. Also try to outsource them to jurisdictions where you can get it faster, because learning about regulations late is pretty expensive in this field, and can, even more than it sets you back monetarily, it can set you back in time, which is even more crucial in this field. So always calculate with regulators from day one.
Perfect. So, if you were to explain in 3 words the work that you do, what would those be?
Expanding extremely efficient therapy.
Okay.
This is kind of forward, but we want to expand this type of therapy to a whole range of new targets, new tumors, and maybe even step into autoimmune disease and other indications in the far future.
Tell us about some of the tools that you use in your day to day life. to become more efficient, or
Ominous AI note taker is kind of your everyday companion. I use a lot of my calendar, and yeah, maybe for the scientific part, a niche software called APE, a plasmid editor, is my best friend, where I edit DNA vectors, try to optimize candidates in there, and yeah, visualize the constructs I want to express before ordering specific DNA strands.
Perfect. So tell us about a book that you love, or a podcast, or any lecture that is your favorite that you keep going back to sort of refresh your memory.
A choose your own lecture module in my doctoral studies, actually, where I went to an immunotherapy special, which is not canon of medical education, but because I was interested in the field, I went there and learned a lot about checkpoint inhibition, which is also a crucial part of immunotherapy, and I predict will be combined with CAR T cell therapy as a standard in the future, because this is what helps stop these escape mechanisms solid tumors have. And CAR T cells themselves, and T cell receptor T cell therapy, which was organized not directly by my medical university, but by the research institute I actually then went to do my doctoral thesis in. So that was pretty nice, and also pivotal moment, because like this is how I got contact to the actual environment I wanted to do the research in. So yeah, and for a podcast, maybe I'd suggest the Molecule podcast, which is of the environment where we have our funding scheme in. So we have a IP token system. We might talk a bit about it later on, for funding the initial research, and it also has a podcast that comes out and presents diverse niche or new projects from the biotech environment, from longevity to bioproduction to cancer projects like ours. And it's really nice to get deep into the biotech rabbit hole if you want that.
Perfect. So, with that, we come to the closure for our podcast. So, we've come to the end of a podcast where Henry shared deeply about the technical aspects of GoCART, cell therapies in cancer treatments. And so, see you next time. Until then, why don't you dive deep into it and understand a little more about life impacting technology and innovation that's happening in the world of cancer treatments? So, see you then.